Hi everyone, today we will learn about another spectroscopy tool which is called as Vibrational Circular Dichroism, that is VCD. In this video, we will learn about what is VCD, its instrumentation and how to record a VCD spectrum. This is our lobby interface where we explain different types of spectroscopy tools. Let's first gather some background about VCD. We all know that when four different atoms or group attached to a single atom, the center is called as chiral and the molecule is called as chiral molecule. The chiral molecule has two configurations. Depending upon the orientation of different atoms or group, it can be assigned as R or S. Both pair is called as pair of enantiomers. To detect the chirality and to assign its absolute configuration, traditional spectroscopy tools like UV, IR, mass will not be helpful. So, characteristical spectroscopy is developed, like electronic circular dichroism, which is also called as ECD and VCD. The ECD works on the principle of electronic transition. The detailed experimental condition for ECD will be explained in another video. There are some drawbacks of the ECD, like the chiral molecule should have chromophore, the spectra is too broad, and less number of peaks are present. To overcome these drawbacks, VCD spectroscopy is developed, which works on the principle of vibrational transition. Here we can see more number of peaks depending on different vibration. In our lab at Niger, we have PMA50 module VCD instrument, which is integrated with Bruker 70B FTIR. Let's go to the instrumentation part of the VCD. We are using the same light source in the VCD which is using FTIR. So the light coming from this FTIR is passes through optical filter. The optical filter gives the measurement wavelength. It is made up of two types. One is low pass filter, another is high pass filter. The low pass filter allows the lower wavelength of light and the high pass filter allows the higher wavelength light. Then the light passes through the polarizer. The polarizer convert unpolarized light to the linear polarized light. Then the light passes through photoelastic modulator, which is also called as PEM. PEM is the important component of the VCD. PEM convert the linear polarized light to this circular polarized light, like left circular polarized light and right circular polarized light. How it is converting? The PEM is made up of zinc selenide material which operates on quarter wave. Due to the quarter plate, there are leading and lagging process takes place. So linear polarized light converted into circular polarized light. Then the light passes through the sample and goes to the detector. Here the sample which is used in the VCD can be solid, liquid or gas. For solid sample, the spectra are recorded by mixing with KBr powder. For liquid sample, the spectra are recorded by using different type of windows like barium fluoride, calcium fluoride, KBr and quartz. Also, we use different size of spacer to control the concentration of the sample. The detector used in the VCD is called as MCD detector, which is mercury, cadmium and telluride detector. We have selected some of the chiral molecules for the demo purpose, like D and L alanine, D and L phenylalanine, R and S camphor, and phenylethanol. Before directly recording the VCD, we first record the IR spectra of the sample. Let's select a chiral molecule which is called alanine. It have two configurations, one is L, another is D. The IR spectra of the L alanine looks like this. It has 5 different peaks in the region of 1560 to 1240. This IR spectra of the L alanine is recorded by mixing the alanine with D2O and in calcium fluoride window. If you will select the IR spectra of the D configuration, the IR spectra also same. Then we calibrate our VCD instrument by with a standard sample and some inputs in PEM, photoelastic modulator. Let's clear the graph and select the PEM. Here we can select a wave number, which region we want to do. Let's say we want to do in fingerprint region, so we can select 1300. If we, it can be vary depending upon the experiment. We can go to CH stretching region, Y stretching region by putting wave number, and the lambda we are using in centimeter inverse, and the retardation is lambda by 4 as we are using quarter plate. If we update this, the modulator frequency will be automatically show 42 kilohertz. Let's see how the calibration curve looks like. The calibration curve A and the calibration curve B. This is the characteristic feature of the standard sample. From this curve, we confirm that the spectrometer is working properly. Then we select our chiral molecule, which is alanine. If we will record the D enantiomer of the alanine, the VCD spectrum looks like the black curve. If we select the L enantiomer, the VCD spectrum of the L enantiomer looks like the red curve. These are the mirror image relationship of the two enantiomers. If you remember the IR spectrum, both has same IR spectrum with equal intensity, but the VCD spectrum is mirror image relationship. So we can assign its stereochemistry by looking at this spectrum. Let's record another chiral molecule, which is camphor. Camphor also has two enantiomers. One is R, another is S. It will record the IR spectra of the S camphor. The IR spectrum looks like this. The IR spectrum is recorded by dissolving the camphor with CCl4 and taken in baryofluoride window. If we record the R enantiomer, the IR spectrum also same. So if we record the VCD spectrum, first clear the graph and record the R enantiomer, the VCD spectrum looks like this. The region we have covered 900 centimeter inverse to 1800. If we record the S enantiomer, the VCD spectrum is just mirror image to each other. So we can easily assign which is R camphor and which is S camphor. These two samples, which is alanine and camphor, is recorded in liquid state. Let's record a solid sample. First clear the graph and select the phenylalanine. The IR and the VCD are recorded by mixing the phenylalanine with KBr powder. If we record the IR spectra of the L-phenylalanine, the IR spectra look like this. If we select the D enantiomer, also the IR spectra is same. And if we record the VCD spectrum of the L enantiomer, the VCD spectrum looks like the black curve. If we will record the D alanine, the VCD spectrum is the red curve. So it is also mirror image relationship. For the IR measurement, the typical scan can be taken 32 or 64. But to get a good VCD spectrum, the scan can be vary from 6000 to 30,000 centimeters. So the last chiral molecule of the today's video is the enantiomer of the phenyl ethanol. Phenyl ethanol also has two enantiomer. One is R, another is S. If we record the IR spectra of the R enantiomer, the spectra looks like the red curve. If we select the S form, it also has same IR spectrum. So if you record the VCD spectrum of the R enantiomer, it looks like the black curve. If you select the S form, it looks like this, the red curve. If you take the concentration of the both the enantiomer is same, you will get the mirror image relationship also with equal intensity. Hope you all understand what is VCD, how to record VCD spectrum, and how to analyze it. Thank you for listening.